Welcome to NewYorkBIO's virtual breakfast series. Digital program started in 2020, bringing you fireside chats with leaders from across the healthcare spectrum. This week's episode features Wayne Koff, the president and CEO of the Human Vaccines Project.
All right. Good morning, everyone. My name is Jennifer Hawks-Bland, and I am the CEO at NewYorkBIO. We're thrilled you're here for another episode of our virtual breakfast series. Thank you to our partners VWR for sponsoring this month. We really appreciate your contribution to our ecosystem, and particularly the discounts that you give to NewYorkBIO members for lab supplies and other services. We have an amazing guest this morning. Wayne Koff will be here to talk all things vaccine. And already we've seen multiple things in the news just this morning about COVID vaccines. So I don't think there's going to be any shortage of conversation topics. Just as a housekeeping reminder, please ask questions throughout our discussion. Don't save them till the very end. Ask your questions throughout our discussion, either in the Q&A box or the chat feature on your screen. Without any further delay, let me hand it over to Derek to introduce our esteemed guest. Thanks.
All right. Good morning, Wayne. How are you? Wayne Koff, founding CEO of the Human Vaccines Project, and you're the chief scientist for IAVI in New York for the better part of 15 years. Would love to hear from you a little more to start off about really kind of how you got where you are and the origins of the Human Vaccines Project.
Sure. Well, it's really wonderful to be here. I spent a lot of time in HIV R&D, HIV vaccine R&D, and at the International AIDS Vaccine Initiative, we had set out, of course, to attempt to make an HIV vaccine. And one of the things we learned really early on is that there are easy pathogens and there are difficult pathogens. And HIV is way over on the right side of the difficult pathogens. Mm-hmm. So after years and years of several attempts of development of a number of vaccines, we took a step back and said that we know a lot about the virus and we don't know a lot about the human immune system. Mm-hmm. And if we had a project that was analogous to the genome project, if you focus on the human immune system, if you could really understand the immune system at the molecular level, it could change everything in the vaccine field. And so, history has taught us that we don't have a vaccine for HIV, we don't have a TB vaccine, or malaria vaccines are really just mediocre. All the cancer vaccines have failed. All the monoclonals against Alzheimer's have failed.
Yep.
So it set out just about 6 years ago and ran a number of meetings across diseases, asked a lot of questions, and spun out the Human Vaccines Project in 2016 as a nonprofit, as a consortium that really is modeled on the Genome Project, with an idea of the decoding of the human immune system as the mission. So that's where we are. And in the last 3 or 4 years, we have shown in essence that the mission is feasible. We have a series of academic partners all over the world. And one of the initial projects was to look at the scale of the immune system. And what I mean by that is every B cell receptor and every T cell receptor. So we went about the comprehensive sequencing of the B cell repertoire and the T cell repertoire, and it gave us a sense of how large is the universe, basically, of the immune system. And what are some of the common elements that we might be able to target to make some vaccines that are 30% effective or 40% effective into 80% or 90% or 100% effective.
Right.
The other issue that we did, or the other set of studies that we did, is we ran a set of studies licensed vaccines, of vaccines that we know work. And we asked the question, can you predict in advance of immunization who's going to do well?
Mm-hmm.
If you look at the virus that we have now in the COVID space, COVID has really shined a light on the bell-shaped curve of the human immune system, and so that the kids and the younger adults are doing, reasonably well with the asymptomatic or the mild disease, and yet it's the elderly that have the disease that is severe in large part, and is causing a lot of morbidity and mortality.
Okay.
So
Do
if
we
we
know?
could
Can we?
understand that, if we could understand that, we could really improve vaccine development across the board.
Yeah. What are some of the things that we do understand right now about the immune system in younger individuals and the immune system in the elderly? And what are some of the challenges of trying to develop a vaccine really for either group?
Well, so if you look at the immune system in total, it's like a U-shaped curve, where the infants and the newborns have immature immune system, and that's why they are prone to several of the diseases, and the elderly have waning immune systems. And
Right.
so it's really the kids and the younger adults have the best immune systems. With that said, we and others are beginning to understand now that there is a chronological age that everybody has, but there's also is a biological age that everyone has, where our markers, our set of markers, are different, and we don't quite understand it yet. But there are people 80 years old that have an immune system like a 40-year-old, and vice versa. And so these are the kinds of the issues across diseases that the Human Vaccines Project is looking at. And of course, it plays into now issues around the COVID vaccine. I mean, there's been this unprecedented effort across the world over the course of a year in the vaccine development space.
Mm-hmm.
It generally takes a decade to make a vaccine. It takes it costs about $1 billion, and most of the vaccines are going to fail. In this case, we kind of got lucky a little bit, in the sense that the virus, the COVID virus, is relatively easy to neutralize, is relatively has a target, if you will, on the outer protein of the virus that is relatively easy accessible.
Yeah.
And it's not a variable virus. It does mutate, but it doesn't mutate on the scale that influenza does, that HIV does.
Yeah.
And such. And so, most of us felt that as a result of a lot of the work that had come out of the HIV field, frankly, where we had the initiation of the platforms that are used, the mRNA platform, for example, that is the basis of the Moderna and the Pfizer vaccines, the vectors that are the basis of the AstraZeneca and the Janssen vaccines, et cetera. It was relatively a situation of a plug and play, which had built on years of the foundation of basic research from the NIH, and years of infrastructure building, from organizations such as the Gates Foundation, NIH and other agencies and such. And so if COVID had come along a decade ago, it would have been a lot harder. And we wouldn't be in a position of the rollout of a vaccine, within a year after the pathogen's been identified.
Yeah. What are some of the? Go ahead.
I was just saying, can we go back to the immune system, sort of, like the chronological age and the biological age? Is that impacted at all in youth by the number of childhood vaccines that we already administer? Or are you talking about just underlying immune system absent of any current sort of structure of vaccination.
No, I'm talking about the underlying immune system in the sense that everybody's immune system is a little different, on the basis of what we eat, what our age is, who our parents are, what we've been exposed to, et cetera, et cetera. And yet evolution has taught us over hundreds of millions of years that there are common ways that the immune system actually works. And we don't understand those principles yet. And it wasn't until about a decade ago that we began to have the tools, if you will. There's been these incredible advances in molecular, immunology and genomics over the last decade.
Right.
But also, as importantly, there have been huge advances in systems biology, AI, and machine learning. Because in essence, the scale and the complexity of the immune system dwarfs anything that we ever had to do in the Genome Project. If you think about the analogy of the Human Genome Project, it took about a decade. It took at least $1 billion or $2 billion, and the scale of the immune system is about 1 billion times as large as the genome. And so we didn't have the capacity at the beginning of the millennium here to even think of sequencing the human immunome.
Right.
And we didn't, and we didn't have the computational capacity to be able to analyze the scale of the data. And now we have that. There's been this convergence, and we're able to do things that people had never even imagined 15 or 20 years ago. So I actually see at some point in time, every baby is going to have a genome sequence, and every baby is going to have an immunome sequence.
Right.
And it's gonna lead us into one, the real advent of the personalized medicine, and secondly, it's going to improve some of the universal medicine in terms of making of the vaccines a lot better than they are now in some of the cases. If you think, I'll just give you one example: the influenza vaccine, which we have to take each year, works in about half of the people, and is a lot worse in the elderly. If we really understood how the immune system sees that virus, it's possible to make a universal vaccine that is gonna work across all the populations, and you wouldn't have to take it every year. So that's where the field's able to get to, and you could say by extension, some of the cancer vaccines work on Alzheimer's, work on MS and diabetes.
So when you say you're trying to either sequence or measure the immune system, I'd like to get a handle on what that really means, because it's there's T cells, there's B cells, there's different kinds of antibodies that people make. There's different kinds of potency of these things, and there's a dichotomy between sequence and function that I don't know that we really know. Can we get that out of sequencing things? So what's really the scope of the measurement that you're talking about? Because again, like the genome, it gets much more complicated than just reading the code.
Right. And so like in the situation of the genome, we are at the beginnings of understanding how the human immune system works. If you think about the analogy of the cosmos, where you look up and you see it's billions and billions of stars. It wasn't until we had the Hubble telescope up there, that we could really begin to peer into the galaxies in ways that people hadn't imagined before. Here in the human immune system, the initial thing that we've done in the Human Vaccines Project is to get a scale and sequence every B cell receptor and every T cell receptor in the blood. That gives us an initial map, and obviously now we have to tease on down because the immune system is complex. It's in organs, it's in single cells, and we have the tools now of single cell, RNA sequencing and other things like that to be able to ask questions that we haven't been able to answer. Here's an example, and the way we have tackled it in the Human Vaccines Project is to use vaccines as probes. So I'll give you a specific example. The Hepatitis B vaccine is a licensed vaccine. It works between 80 and 90% efficacy. And so as an experiment, in some people, in the majority of people, it's a 3 immunization regimen. It's given at 0, 1, and 6 months. A quarter of the people are protected after a single immunization. 10 or 15% of people are never protected. So we have an experimental system, a human experimental system. And so we immunize individuals with the hepatitis B vaccine, or we've also done influenza and shingles and yellow fever as examples. And we probed the immune system pre-immunization and post-immunization. But we also do an extensive array of the omics assays, of the transcriptomics and proteomics and metabolomics and lipidomics, et cetera, and epigenetics. And we go into tissues. We have the fine needle aspirates of lymph nodes and marrow. And so you begin to see a comprehensive analysis as to how the host receives the vaccine. And in this comprehensive understanding of the immune system, we are then able to generate a series of predictions prior to immunization and models as to who will get the highest titer, who will be protected after a single immunization, and why. So those are the kinds of studies that are moving us along, Derek.
Yeah, so we have a couple of questions here that, delve more into what happens with different tissues and what happens with different cells. One of them is, asking how much of this do we understand that either goes into the brain or the CNS, but you could also talk a little bit about how we know whether it's protective in certain kinds of tissues or different kinds of conditions if we just start with the blood.
Well, so let me give you just an example of some of the unknowns, and it really applies into COVID. The major question on the table, or major two of the questions on the table right now for the COVID vaccines are really how long they're going to last in terms of, of protective efficacy, and are they going to work as well in everybody?
Right.
And so we don't have a biomarker at the present time for how long a vaccine is going to be effective. But there likely are biomarkers. We just haven't identified them yet. And
I was going to say, how are we going to know? Like, are we going to know the duration only by retrospective analyses?
Well, right now, that's the only way we're going to be able to do it.
Yeah.
But at the same time, we're in discussions now with all of our partners, all of our industry and biotech partners, who are the makers of the leading COVID vaccines, and they all understand that these are the two of the principal issues. And so it's likely that while the vaccines are rolling out, that there are going to be a series of vaccine trials of small, hundreds of people who are comprehensively analyzed in a vaccine efficacy trial. In 60,000 people that we use in one trial and 40,000 in another trial, that you don't really have the capacity of taking all the samples that you want to take. Yeah. Here you could do a trial of a couple hundred people and intensively monitor them and look at pre-immunization markers as we've done for the Hepatitis B vaccine, where we identified markers of who is protected after a single immunization and who's going to make the highest of the titer, that you could begin to run studies in like this with a COVID vaccine, or for that matter, other vaccines, because in this case, it should be platform specific and not antigen specific, and identify what the markers are. So it's a comprehensive analysis. It's a systems biological analysis. And where we really want to go with this at the end of the day, and this is gonna take a longer time than I'm gonna be working on vaccines, certainly, is to create an AI model of the human immune system.
Yeah.
So when we think about measuring the human immune system, is one of the challenges just the type and amounts of sample that you have to take? Because if we're looking at things like functional T cells, they're relatively rare, right? So you end up having to take a pretty decent amount of blood, and you end up having to process it. So you're actually looking at at the actual immune cell layer. Is that one of the hurdles that we're actually thinking of here, or that we're facing?
It is, it is one of the hurdles, but it is a surmountable hurdle. A lot of the assays, and we've done vaccine trials in infants, and so a lot of the assays now are miniaturized, and so you can really do a lot more with a smaller amount of blood than you ever could before.
Okay.
With that said, some of the functional assays, you still want to take larger volumes in a subset of people.
Yeah.
The other point about the immune system is a lot of the action occurs in the tissues, and some of the markers can be identified in the blood, but other markers are only going to be identified in tissues. And so in the vaccines project, we basically pioneered the work of some of the fine needle aspirations of lymph nodes and marrow to be able to look at what is occurring pre-immunization and post-immunization in the organs that actually matter.
We actually have a question about we've had several questions sort of throughout our breakfast series about IP, or intellectual property. And the question is, the use of markers is an important advance. Have you or your partners in the Human Vaccines Project encountered difficulties in securing patent protections for the new techniques? How are you all handling IP in the work you're doing?
Yeah, that's a great question. So we're working in the pre-competitive space in the Human Vaccines Project. But obviously there are a lot of applications that are coming out of the data.
Yeah.
And so we have patent applications actually on some of the predictive markers. The patent applications are moving through the process, so it's a little early to say. And we've had a number of the companies outside of the project who are not the corporate partners at this time of the project, who've approached us for one of the database or another database or whatever. It's a tension in a sense that we want to open source the data, yeah, as fast as possible and let the whole research community look at it. On the other hand, there are specific applications that are going to come out. One of the hypotheses that has been generated already in the first couple of years is that the baseline is predictive of outcome, which is not a novel, hypothesis, not a novel idea, in its own right. However, we're beginning to understand what is baseline. And as we understand more and more about what is baseline, we then have the opportunity of the modulation of baseline. And so it's possible, Jennifer, that as we go along, that one can think of an adjuvant or a vitamin or something else that is used in advance of or with a vaccine, which is going to increase the potency or the durability of the depth, or whatever.
Mhm. So we had a question earlier about adenovirus-based vaccines, and this person asked about the AstraZeneca trial and the half dose showing a higher efficacy than the full dose. So the other piece of the question was, what do we think about adenovirus-based vaccines if this becomes actually a yearly type of occurrence? Can you talk a little bit about kind of that modality, because this is a modality that's been around, that's been developed over the last little while, and this is now just being kind of put into the COVID space.
Yeah, sure, and that's another excellent question. The adenovirus vaccines as a platform have been around a while, and certainly a lot of work in the HIV and TB and malaria vaccines are based on adenoviruses. And they really are an excellent system in terms of expression of a foreign antigen. And as we've seen, induction of neutralizing antibody responses and cellular responses. There are some adenoviruses that are better than others, and what I mean by that is that we've all been exposed to a lot of the adenoviruses that are the causes of the common colds and sore throats and whatever. And so one of the challenges for adenoviruses is something known as anti-vector immunity. Mm-hmm. And so if you've got antibodies against the adenovirus, or if you've got immunity against that type of adenovirus, it's difficult, or it can be difficult if you came in with the same of the vector. And so AstraZeneca, as an example, and the team at Oxford that has done the research on the chimpanzee adenovectors, mm-hmm. has chosen a vector, a chimpanzee vector, as a result of a way of getting around anti-vector immunity. It's possible, though, that some of the vectors after a single immunization are going to induce mm-hmm. enough anti-vector immunity that you may not be able to give a booster.
Yep.
And so Janssen, as an example, has another vector, which they're using, and they're looking at a comparison between a one-shot vaccine and a two-shot vaccine.
Right.
And if the one-shot vaccine, is effective, it's a game changer.
So should we be worried now about the two-shot vaccination regimen? Do you think that's a very large hurdle to overcome in terms of distribution?
Well, it's not a very large hurdle, but it's going to be a hurdle, because I was just on a webinar the other day where a number of the clinicians that have immunized kids over the years, where they have 3-shot vaccines like the hepatitis B, there is a compliance issue.
Yeah,
and more people show up after the first one, and less at the second, and less after that. So I think we're going to have. Yeah, I think we're going to have to do an education, campaign here, which gets into another issues of the vaccine field. I mean, I think achievement of the efficacy of the vaccine is obviously is a major advance, but the real advance is vaccinating the public.
Yeah.
And in the case of the COVID vaccines, we're in a new world, in the sense that historically we've made tremendous advances with work by agencies such as the WHO and UNICEF and Gavi on the deployment of vaccines, and the percent of infants and kids who are receiving vaccines increases every year.
Right.
In contrast, we don't do as good of a job on the vaccination of adults and elderly. The influenza vaccine only hits about one half of the population.
Mm-hmm.
So we have to learn how to do that while we're doing it. We have to build the ship and sail the ship, if you will, at the same time. And so this is going to be a logistic operation, unprecedented in scale, in the sense that if we want to achieve the population of based immunity across the world, then we're going to have to immunize, 60, 70, 80% of the world.
Right.
Yeah, I think everybody is already accustomed to bringing their kids to get vaccinated by and large, and there really hasn't been a time where, as an adult, you yourself and the other adults that you're around also have to go and get vaccinated. We've been kind of phased out of that routine medical procedure type mentality, right? I would imagine that at this point, it's almost kind of like we have to have a we're all in this together type of spirit with this. But like you said, communicating around that is going to be difficult. There are some people that aren't going to want to get on the boat.
Well, yeah, and certainly, and obviously on this call we don't want to get into a lot of the politics, but the reality is that over the last 3 or 4 years we've seen an assault on science, and we've seen an assault on facts, and the polls indicate that only 60% or so of people in this country want to take a COVID vaccine, and that isn't good enough.
No.
And so we're going to have to do a lot of work in that space in the coming months.
Well, and two, we have a situation now where obviously the states will individually the federal government will provide the vaccines, right? And the states, by and large, determine the distribution within their jurisdictions. So then that also leads to a lot of, different states are different. Obviously, like Wyoming has a very different distribution system than New York, where, we're very much more densely populated. But I think that's going to be a challenge too, is just figuring out where people get vaccinated, and particularly with the 2-dose regimen, making sure they go back to the same, like how do they figure out where to go back and when to go back and following up? I think that'll be a challenge.
Absolutely. And if we just look in the country, and the examples of Wyoming and New York are perfect in a sense that there's a difference in the outlook on the disease. There's a difference in terms of levels of social distancing, wearing masks, etc. So we've got a long way to go here in terms of education of the public in a short amount of time, because these vaccines are likely going to roll out at the end of the week. The FDA is meeting in this week on the potential of the emergency authorization of the Pfizer vaccine. The Moderna one will come right after that, and this is one of the other challenges that we face.
Do you see any window for, say, follow-up testing as part of the kind of communication compliance thing here, and then that will actually help us measure the response?
Well, I mean, I think, and I know there is going to be a lot of work on attempting to identify what is the biomarker of the correlate of the protection. Mm-hmm. So a lot of research is going on in that phase. It looks like it's going to be an antibody marker. It might be neutralizing antibody. It might be a level of neutralizing antibody. But the issue of the contact tracing and the follow-up tracing is just a whole other issue there, where I understand the latest data is we do a couple of million tests a day in this country now, but that still is a lot less than what we're going to need. And when you have a couple of 100,000 of the cases each day, as we do in the last week, it makes the issue of the contact tracing even more challenging. So these are real society challenges in addition to the research and development of the vaccine.
I mean, has anybody proposed having either an antibody test or something like it as part of the follow-up? I mean, one could imagine if you have a 2-stage vaccine booster, you could almost have a a before an initial and then a month after the second boost. I mean, I realize that brings you now up to kind of 3 visits or tests per person, but at some point we have to think about what's really the best way to measure this going forward, because the thing that I think we're going to be reliant on is data, and you can't retroactively go back and say, oh, we wish we had this data here if it was going to be really useful. So one of the challenges, I think, is communicating which data we absolutely need on the people that get vaccinated, or on a population scale, rather than a million tests a day. And is a test a rapid test? Is it an antigen test? Is it a PCR test? Is it an antibody test? All of these are different flavors of ice cream that don't do the same thing.
Right. It's a fluid environment. It's a moving target, and people have raised issues of do we need an antibody test as a passport, and do we need an antibody test to be able to open up the country, and other things like that. And one of the reasons that isn't going to work is that we don't understand the durability of the protection, and we don't have the biomarkers. And so I think the initial thing to do is to roll out the vaccine or vaccines, because it looks like there are going to be a number of vaccines over the next 3 to 6 months, basically, that will be licensed. I think we need to roll those out. We need to really look intensively in the post marketing of the vaccines in terms of levels of safety, any other additional adverse reactions. It looks like, from all the data that I've seen, that these vaccines are extremely safe, which is really excellent. And then we need to, in terms of the R&D effort, we need to attempt to ensure that as many samples are taken in the vaccine trials, and the right samples are taken at the right times, to enable organizations like the Human Vaccines Project and the Human Immunology Consortium that the NIH supports, and other of related organizations like that, to really probe, because it's going to help us on the next epidemic, and it's going to help us on the next pathogen as we go ahead. I mean, on the one hand, it really has been an unprecedented effort, to be able to go from identification of a virus and sequencing of the genome of the virus to hundreds of the candidates in development, and tens of candidates in vaccine testing already, efficacy sheen, and a rollout of the vaccines within a year. But even with that unprecedented effort, we have 60 million infections, or 60 million cases.
Right.
1.5 million have died. Those numbers are still going to go a long way up over the course of the year.
Yeah.
And the real question is really, how do we get out in front? Is there a way to make a universal coronavirus vaccine? Is there a way to make a universal flu vaccine? Is there a way to get over the hump with the HIV vaccine? Well, obviously, I've got a bias here, and the bias is we have to understand the human immune system a lot better than we do now. But I think we have the tools to be able to do that.
So in addition to the No, go ahead, Jennifer. That's okay.
How do we, and something you said just made me think of this. How do we avoid multiple vaccines? How do we avoid this idea that people fixate on one vaccine? Like, I want the Pfizer BioNTech vaccine, or I want the Moderna, or it's not exactly, it's not like, Campbell's versus Progresso or whatever different types of soup. You get to go to the shelf and just pick your favorite. How do we address that when we're talking with sort of the public at large?
Yeah, that's a great question. I mean, I think, well, I'll give you an analogy. As I'm over 65 now, I went to see a doctor a couple of years ago to get a flu vaccine, And the doctor asked me if I wanted the high dose vaccine or if I wanted the regular dose. And so I said, well, that's an interesting, that's an interesting. Sorry. That's an interesting, interesting idea. And I've worked on flu vaccines, et cetera, et cetera. And we got into a discussion about that. And I walked out the door and I said, the next patient that comes into the doctor's office is a carpenter or is a plumber or is a teacher or
Right.
whatever, and has no knowledge about vaccines, and it's not going to be able to make a decision.
It's not the same as would you like another shot of espresso in your cappuccino,
That's
right? It's not the same thing.
Right, exactly. So and so I think we have to educate the public, and the practitioners have to be educated also, because I don't envision any scenario where you're going to walk into the doctor's office or the pharmacy and you're going to get a menu. You're going to get a vaccine.
Oh,
and
yeah.
you're going to get the vaccine that they have there at that time.
Well, even when my kids were little, right? I worked at Merck, right? I would ask, which vaccine are you giving my children? It wasn't like I had the Merck vaccine sent to the doctor's office, but they would look at me like, why are you asking this? And I was like, oh, I'm just curious. Like, I just want to know. Yeah, but I think you're right. But I think doctors need to be able, I think physicians and those that administer the vaccine need to be able to explain to a patient, and even if it's just like, this is the vaccine that we've received in our state, this, like that we need to prepare them with the right facts, I think.
Yeah. And, and I think in this country in particular, and it certainly is similar in a lot of other countries, but in this one in particular, we have a pretty strong anti-vaxxer movement.
Yes.
And we have to address that really head on here, or we're not going to achieve the levels of protective immunity that we need.
Yes.
We want.
Right.
And so I know there's a lot of people, that understand that area a lot better than I do, and the whole education arena and sociology associated with that. But we have tools now to identify, how safe vaccines are, and we are beginning. We don't have all of them yet, but we're also beginning to have the biomarkers of safety. And so, as the vaccine field evolves over time, I really envision a point where we're going to be able to get over the hump on a lot of these diseases that until now have been intractable, and in particular in the non-communicable diseases, where the elderly are facing, increasing numbers of autoimmune diseases, the neurodegenerative diseases, the cancers, etc.
So we had a question earlier that I think kind of fits in now, since you just mentioned this. Somebody had asked about efforts to develop T-cell based vaccines for COVID, but also cancer and other modalities. So can you talk a little bit about vaccines, say in the cancer space or in, I'll say, non-infectious diseases, and what kind of some of the biggest hurdles that they're facing are, and really what you think the outlook is for some of those things, given the advances that we've seen?
So let me give you like a 60-minute, 60 seconds. Sorry.
Plenty of time. We got plenty of time left.
Seconds. Yeah, I can use another cup of coffee. But in simple terms, if you look at the two of the major arms of the immune system, the antibodies are going to attack a virus and neutralize it. The T cells are going to attack the infected cells and eliminate them. And so the ideal vaccines have a combination of the antibody and cellular immunity. The immune system is a surveillance system. So, in terms of cancer, the immune system is seeking out all the time anything inside the body that looks like it shouldn't be there, is foreign. And so it's, if we get infected with a virus or a bacteria or with a parasite, the immune system obviously sees that as foreign. In the case of the cancers, it's the T cell arm of the immune system that is the surveillance arm. And so it's looking at healthy cells or normal cells. It's looking at precancerous cells, and it's looking at the cancer cells. So all of the cancer vaccines to date have basically failed. And part of the reason that they've failed is that we haven't understood enough about how to marshal the T cell arm of the immune system in vaccines to be able to see those precancerous cells and early cancerous cells and wipe them out. And when you get later and later on into the disease, there have been huge advances. I mean, the Nobel Prize a couple of years ago to Allison and Hanjo was to really elucidate the idea that there is a break or a series of breaks on the human immune system, PD1, CTLA4, etc. And when you unlock the break, you unlock the immune system.
Yeah.
But it also turned out when you unlock the break, you can overheat the immune system, and so it's a balance, and we haven't quite understood exactly yet really how to get that right.
Yeah.
But we have the tools now to be able to do that. And so we're actually looking now in the cancer space to begin a series of studies with one or more of the experimental of the cancer vaccines, with or without the PD1 inhibitors. These kinds of studies where you can comprehensively analyze, where you're able to take a lot of samples and begin to understand what are the predictive of markers there that we don't understand yet. Yeah. So it's coming, but it just isn't there yet.
Yeah. I imagine, if you embark on, the understanding of the whole immune system with regard to something like vaccines, eventually you branch off into understanding hyperimmune responses like cytokine release that are, now, big challenges in the CAR T space. And, you think about septic shock, you think about all of these other things that are extensions of, challenges against the immune system in terms of a preventative thing, as opposed to just, a surveillance aspect.
Yeah. I mean, it's interesting in the hepatitis B work that we did. Some of the predictive markers that we identified were on the innate immune system and not on the adaptive immune system, and that whole complex area of cells that are the initial kind of signaling cells for the adaptive system to be able to take off are not understood as well. But I think it may turn out. What we are learning more and more is there is a bell-shaped curve of the human immune system, and so there are some people who never get a flu that is a bad flu, and there are other people who always get a cold that is a terrible cold, in really simplistic terms. Well, if we could understand, what are the low and medium responders and high responders, we should be able to modulate the curve and move people from low to medium and medium to high. And this gets into, by extension, why are a subset of people are going to get either cancer X or cancer Y? Why are a subset of people are going to get Alzheimer's? Why are a subset of people are going to get a severe, infection with COVID? This is the bell-shaped curve that I think is the next frontier, frankly, of human medicine.
Yeah. So do you think we're going to get to the point where, you know, people individually are going to understand their immune systems enough to change their behavior? I think some people with chronic conditions do this a little bit now, but, you know, are we going to get to the point where we actually are going to be able to think about, you know, what does our immune system look like on an individualized basis and use that to better make decisions about, you know, what kind of things we do and how we do them?
Absolutely.
Yeah.
I mean, I think at the end of the day, you know, and it's going to take us years, but at the end of the day, given with the advances in the technologies of human immunology, systems biology, and AI, I think we are going to get to an AI model of the human immune system. And I think that people, you know, a generation from now are going to have their immunome sequenced as a routine. And you know, if you think about it now, if you go into your doctor's office annually and you're going to get a physical, if you ask the doctor, how's my immune system, he looks at you like you're an alien.
Yeah.
He has absolutely
Yeah, I don't know.
Either he or she has no tools to really be able to look at that. Well,
Right.
that's all going to change. That is absolutely going to change.
Because right now, even our freezer went out over the weekend, and my repair person told me about all the supplements he's taking to boost his immune system, right? And so, you know, and it's just like some of it's guesswork, even working with his physician, but like some of it's guesswork, you know?
Absolutely. And unfortunately, that plays into how well that people are doing or not doing with one vaccine or another vaccine.
Yeah.
So I'm really excited about it. I mean, I think that you know, this is a renaissance period in terms of vaccinology, and it's a renaissance period in terms of immunotherapy, and it behooves us as a field to really, you know, recognize that we're in this period and make the most of it.
Do you think that COVID is, I mean, clearly you said it's a renaissance period. Is it bringing more attention, i.e. research dollars, into looking at immunology now?
I think it will. I think the billions of dollars that have come in, and rightfully so, have gone to let's get a vaccine as soon as we possibly can.
Right.
But I think the unique opportunity that COVID has brought us in the research space is that we have a similar antigen, the S protein, the outer protein of the virus.
Yeah.
In mRNA vaccines, in DNA vaccines, in adenovirus and VSV vector vaccines, in killed vaccines, and in live attenuated vaccines. So we really have an opportunity as a field to undertake these analyses of the platforms and the comparative analyses of the platforms. We may never have another opportunity that is like this. And so hopefully the resources will be there. And if they are there, I think we're going to make tremendous advances in the next decade. I certainly think that within a 5-year period, that, you know, with enough of the resources, we could understand the human immunome as we understand the genome, you know, now, you know, as much as we understand the genome now. I think within a decade, we could understand, you know, what are the rules of effective immunity. And I think just after that, once you have all that data, which is a huge amount of data, it's going to feed the artificial intelligence industry to be able to come up with initial models of the human immune system and to iterate on those models as we get more and more of the data. If you think about the old weather models and the predictions of the hurricanes and where we were a couple of decades ago and where we are now, I think that's where we're going to get to on the human immune system.
Yeah. So we've had a couple of questions about this, and I think this is probably a good one to close out on. So I think one of the biggest things that everyone is going to ask over the next few months is, you know, when am I going to get a vaccine, right? And, you know, if you had if you had kind of power over how to communicate with people, what would be some of the things that you would try and message to people in terms of when their group might be vaccinated? What are some of the big kind of communication strategies you would think of for a wide range of people in the entire country that's really all concerned about the same thing, which is when do me and my family get a vaccine, and when do we get protected? What are some of the ways that you would try and do that?
Well, I think the most important aspect of the messaging is going to be to educate the public really on how safe these vaccines are.
Mm-hmm.
I think the major obstacle in the minds of people who are reticent to take the vaccines, who are reticent to either give a child a vaccine or give it to an elderly parent, mm-hmm. Are the vaccines safe? And so I think we have to hammer home the message that the vaccines are safe. Mm-hmm. I also think we have to hammer home the message that in addition to the individual, it's a public good. It's a societal message. If we don't all get on board the ship and row in the same direction here, we're not going to get there. Yeah. And so if somebody's offered the vaccine, they should take it. Yeah. And the rollout of the vaccines, obviously, as they're beginning to roll out now with the high risk of workers and the elderly and others, but as more and more vaccine is made, the vaccines are going to be rolled out in series into different population groups. We need to hammer home that message of safety, efficacy, and community.
And Bio at the national level, and we are going to link to this on the NewYorkBIO website, has come out with a new website just over the weekend that's covidvaccinefacts.org, and that dispels a lot of the myths around and talks about how vaccines were developed, how they've been safely developed, and what the process is. So we'll be pushing that out as well to make sure that the public and policymakers can understand that the process was robust and it was a serious process. It wasn't something that companies just threw together. It was very much in line with our standards.
Yeah, no, I think that's absolutely right. I think the other thing that the public has to understand is that there isn't anything that is 100% safe.
Right.
So that there will be adverse reactions, and whether it's 1 in 100,000, 1 in a million, 1 in 100 million, we don't know that yet. But overall, if you balance the importance of the protection in getting the world on a track towards whatever the normal is going to be,
right?
I'm all in.
Good. Well, that's the perfect ending. Yep. Wayne, thank you so much for being our guest today. What a fantastic discussion. Lots of engagement from the audience. We really appreciate your spending this hour with us and with our stakeholders. So thank you.
Well, thanks again. And you all go to the Human Vaccines Project website, see what we're doing. And we need a lot of help, and we need a lot of support. We need a lot of partners. And so I've enjoyed it. I hope you've enjoyed it. And now I get a little breakfast.
Yes. Thank you. Thanks, everyone.
Right. Released. Thanks, Wayne. Thanks, everyone. Have a great day.
Sure. Take care.
Thank you for tuning in to NewYorkBIO's virtual breakfast series. Join us every Tuesday at 9 a.m. for more discussions with leaders from across the healthcare spectrum. For more information on NewYorkBIO, please visit us at www.newyorkbio.org.